That's right. I think you'd be really, really wealthy if you got pregnant anyway.
I think you might be. You could figure that one out. I would pay you.
Mm-hmm. You'd be a surrogate.
There's a girl in my biz lab who's like, I just love being pregnant. I have 3 kids, and I love every minute of it. I'm like,
Yeah, I never felt like that. Mm-mm. I like the kid. He's awesome. How are you? Good. Oh, you are? I have a friend who really, really wants to be a surrogate for a couple. What? And then for private?
donate eggs.
Donating your eggs to fertility clinics privately in Utah. It's going to be $10,000.
Oh my goodness.
I know, I looked into it. Put you on a bunch of like hormonal drugs.
You don't need that, honestly.
No, I know.
You are too delicate. Your system would, like... She is delicate.
Tell me, it's not worth the money.
Well, I'm like, let's work, but
Maybe the girlfriend do that so she gets to my college.
No.
at least like financially it's stable.
Okay.
That's true.
You guys ready? Ready, Freddies? You get ready. What are you doing back there?
Can't sit up front.
Oh, okay. I thought you guys had spat.
No.
Not yet. Lover's quarrel. Brother and sister quarrel is more like it. Okay, so I can't remember. We did autocrine agents. Yes?
We were at
These guys?
a little bit.
Okay, I'm just gonna like briefly. Okay, so I did this time. The last recording didn't work. I don't know why. Oh, let me turn this thing on too. I always forget. There you go. Okay, so is there ever a situation in which you don't have to use an intercellular chemical messenger to communicate between two cells? And the answer is yes, but it's very rare. The two cells would have to be right next to one another, right? And they could be connected by gap junctions. And then you can communicate almost instantly without ever releasing anything into the extracellular fluid, right? Okay. So why not use gap junctions? Well, first of all, not all cells are next to each other. And then second, gap junctions are kind of a one-trick pony because that means that you would have to do exactly the same thing as the cell before you. Whereas other cells can have like 100 different kinds of receptors, and you could tell them turn on, turn off, do apoptosis, go through cell division. So they can take a bunch of different commands. But with gap junctions, the cells right next to one another are going to do pretty much the identical things. So gap junctions are good for almost instantaneous communication, but they're not very varied. The communication cannot be varied. So they're not very common in the adult body. You have them, you probably remember from anatomy that you have them in your respiratory epithelium to wave all that junk up. You have them in cardiac muscle cells and smooth muscle cells, not very many places. Interestingly, before your nervous system was mature, when it was an embryonic and fetal nervous system, putting together the neurons, difficult proposition, making sure that things are connected to one another properly. Does that make sense? Like getting everything connected properly. So it seems as if the way the embryonic brain develops is that everything is put together sort of with gap junctions so that you've like got the structural stability. You don't yet have the ability to release different neurotransmitters, but then eventually, as you've already got the structure down, the gap junctions pop apart and then they start communicating with neurotransmitters. So the embryonic and fetal nervous system has a whole bunch of gap junctions, but as adults you don't. Okay, so that is gap junctions. Your textbook, for some reason, calls gap junctions connexons. I don't know why. That textbook just likes to use that term, but I don't ever use that term, and they don't use it in anatomy either. Okay, so see a chemical. How do you know whether it's a neurotransmitter, a hormone, a paracrine or autocrine agent? How do you know what it is? Well, you don't memorize it. You don't say, hey, I know dopamine. Dopamine's a neurotransmitter. Because once your body figures out how to make something, it can reuse it any damn way it pleases. For instance, dopamine is a neurotransmitter very often in the brain, released between two neurons in the brain. It also, however, gets dumped into the bloodstream sometimes, and then it behaves as a neurotransmitter. As a hormone. So instead of memorizing serotonin is a neurotransmitter, acetylcholine is a this, then you actually look at what it's doing. Because you can and will see things reused in the body. And it seems counterintuitive, but once your body figured out how to make something, there's no reason it can't use it for multiple purposes, right? As a matter of fact, believe it or not, ATP, which of course you guys all know is an energy molecule, is also used as a neurotransmitter in the brain. Because like, hey, I already know how to make ATP. How about I just stick it out there and let it turn something on, and then I'll pull it back in and use it as energy. Right? So you look at what it's doing. The single molecule could be any of them. You have to look at what it's doing. Okay, and that's it for that set of notes. And now we will start on chapter 2 and 3, which is cell structure, function, and metabolism. I don't take a whole lot of time here to go through what you spent a lot of time on in 1610, which is what's a ribosome and what does all this crap do. I'm assuming that you will know that, and we're going to start with Roman numeral 2. And we're going to start with metabolism, which is chapter 3. And we've got to approach metabolism from a physiological perspective. So the idea here is when we talk about metabolism, a lot of times people are talking about, okay, why that guy can eat like 2 cheeseburgers and never gain a pound, and you eat a cookie and gain 3, right? But what we're going to try to do is approach it from a cellular perspective and then extrapolate to a whole body perspective, right? So we're going to say that metabolism is collectively all of the chemical equations that happen inside a cell, all the chemical reactions, both the reactions that give off energy and the reactions that consume energy. Does that make sense? Okay. I want to say produce and consume, but, you know, the laws of physics say that energy is really never created and it's not destroyed. It is used and generated. So it's collectively all of those chemical reactions in a cell. But if you take all the cells, of course, metabolism can include such things as if you are doing more energy producing reactions than you are doing energy consuming reactions, then you will store the excess. Not at a cellular level, but at a body-wide level, you will. Does that make sense? Go ahead. We already talked about that. Yeah. Okay. So at a body-wide level, you actually do have the capacity to store. At a cell level, not so much. But the metabolism can be broken down to 2 sort of subcategories, and they are anabolism and catabolism. You guys match up to the descriptions with the terms. One of those 2 terms is generally taking small things and building them into larger molecules at the expense of energy. Anabolism, exactly. And catabolism is generally taking large things, like, for instance, glucose, and breaking it down to smaller things like carbon dioxide and water and giving off energy, right? And so generally speaking, what happens inside your cells, it's that you couple catabolic and anabolic reactions so that one of them drives the other. Okay, and you will actually see these terms in other contexts as you go through physiology. For instance, testosterone is considered an anabolic steroid because it causes you to build muscle mass. Yes? Does that make sense? But cortisol is considered a catabolic steroid because it actually causes you to degenerate your muscle mass. Does that make sense? So those terms will come back again. All right, so that's collectively what we're going to be looking at. And then now let's just start talking about enzymes a little bit. So with chemical reactions inside cells, not inside a test tube, generally there are a lot of enzyme catalyzed chemical reactions. So the chemical reaction will occur more quickly and more effectively in the presence of an enzyme than in the absence of an enzyme. So the way that they usually describe this, for some reason, doesn't make a lot of sense to most people. They describe enzymes as lowering the activation energy for a chemical equation or a chemical reaction. The problem is that the concept of activation energy doesn't mean anything to most people, especially if you're not dealing with an energy requiring reaction. They're like, okay, you just told me this reaction doesn't require energy, and now we're lowering the activation energy, and why the hell are you trying to confuse me? Well, the deal with enzymes is they're really not that difficult to understand if you just think in terms of probability and statistical likelihood. So this is the way I think of enzymes. And it is true, and it does end up accomplishing the same thing. So if I have a chemical equation that I want to occur in a cell, and it's just a simple chemical equation, we don't even have to use real chemicals. This is what I want to occur. I want A plus B to go to A bonded to B. And A has a specific shape, and B has a specific shape, and A bonded to B would look like that. So I could just, inside a cell, wait for them to sort of run into one another, right? And then you would get that equation or that reaction every once in a while. But what if I provided a location for them where they were attracted? This location is your enzyme. And now I have a location that's attracted to A and attracted to B. Can you see how much more likely that chemical reaction is going to occur if you provide a location that has an attractive binding site for A and an attractive binding site for B? So it's just statistical likelihood, isn't it? Like, and if you have more enzyme, are there going to be collectively more of the product? Yeah? And therefore the speed of chemical reaction is going to increase as you add more enzyme. Does that make sense to you guys? So this is what enzymes really do. They just provide a location for a desirable chemical reaction to occur. They're not even really involved in the reaction, right? They're just the place that you meet up. So if your chemical reaction that you wanted to occur was you want to meet a nice girl to take home to mom, then you go to church, enzyme, and you're more likely to find your B, right? That's what you're looking for. However, if you want to hook up, you don't go to church. It could happen, and it has, but you go to the bar, right? Different reaction, different enzyme, right? Does that make sense? So it's providing a location where things are going to occur. And so if you're looking at an enzyme-catalyzed chemical reaction, there's some words for these. Anytime you're looking at a unidirectional chemical reaction, whether it's enzyme-catalyzed or not, these are reactants, right? And these are products, yes? But when you're looking at an enzyme-catalyzed chemical reaction, the reactants also are considered substrates. And they fit in the enzyme at these sites, which are called active sites. Active sites, by definition, only fit the substrate. Is that okay? Now, do you understand what I mean when I say the enzyme isn't actually really involved in the process? It's just like a location for it to occur. Do you see also why it can be reused? The enzyme can be reused, and it is reused. So they lower the activation energy for a chemical reaction or catalyze the chemical reaction, but you can just look at it as statistical likelihood. So the more enzyme you have and the more substrates you have, the more your chemical reaction is going to go up. Does that make sense to you guys? Okay. All right. So the substrates combine with the enzyme's active site, and the enzyme is, of course, not consumed or changed permanently in the reaction, so it can be reused, but it's not like you made all the enzyme you were ever going to have as an infant and you're still using it. I mean, it wears out, just like everything, right? I mean, you don't need to buy a car every single time you need to drive to work, but you buy one and then 10 years later you're going to have to buy another one, right? Because it is eventually going to wear out. So your enzymes do have to be replenished eventually. So if you could not make any more, you'd still be screwed, right? You would basically have run out of the ones from when you were a kid. How long they take completely varies with the enzyme and how often it's used. Okay, so what we want to look at now is if I'm talking about an enzyme-catalyzed chemical reaction, what kinds of things can impact its speed, the rate of the chemical reaction? And some of them are really intuitive and you would already understand them, but some of them aren't. So let's talk about, let's see, enzyme and product concentration for a second, or enzyme and substrate concentration. Okay, so let's look at, actually let's look at this one first. This one first. Okay, so simple graph, substrate concentration increases, reaction rate increases. Does that make sense to you guys? Okay, if you increase the concentration of the substrate, A and B, then you can get an increase in reaction rate until something happens. What is the something? What's that called? It's called saturation. It's called saturation, okay? Now, saturation is physically something happening with the enzyme. What is physically happening with the enzyme at saturation rate? all of the enzymes are full. And do you see how it limits the rate of the chemical reaction? It doesn't take it down to 0, but it limits it. Because what's happening when you get to saturation is that all of the substrates are just kind of waiting in line for their turn, right? And so you can't go any higher than that. Does that make sense? Even if you add more substrate, you're still not going to get any higher than that because the enzymes are all full. Does that make sense? Okay, so basically, with substrate concentration, as substrate concentration increases, reaction rate tends to do what? Increase until you hit saturation, which is this guy. Right here. Okay, so what is the only way that you can think of that you could actually increase the saturation rate? Exactly, you can increase the enzyme. So if you increase the enzyme concentration, then you can go from this curve, which is substrate concentration. Still, as substrate concentration increases, reaction rate increases until you hit saturation. But if you actually increase the concentration of enzyme, you can increase the reaction rate still, right? Is this instantaneous inside a cell, increasing enzyme concentration? No, it's not instantaneous inside a cell, right? And sometimes you don't necessarily want to do it. You're just going to deal with the enzyme. However, if, for instance, you had dealt with, like, not having enough product for a very long time, your body might need to say, oh, well, maybe I need to adjust the ideal rate of that enzyme or the ideal amount of that enzyme, the set point for that enzyme concentration up. But that's not an instant fix inside a cell. Okay, so you can increase saturation rate, but only if you increase enzyme concentration. Is that good? Okay. All right, so we did substrate concentration, saturation. Okay. All right, so let's talk about these two things. So these two things get a little confusing to people, so make sure you just sort of hold what I'm saying in your head. So this one is called substrate specificity. Okay, so we always think of enzymes as having one substrate, but it's not really true. Enzymes have an ideal substrate, and then they have one that will work in a pinch and one that no way, no how, right? So this is this enzyme's ideal substrate. It is the most specific fit for that enzyme, okay? This is the one that is never going to accept no matter what. This one might do in a pinch, okay? This one, I don't know, it'd have to be a pretty slow day for that one to do, okay? All right, so what do you guys think? The better the fit, the more specific the fit to the enzyme, the what the reaction rate? Higher the reaction rate. Because it's going to be more likely to fit, right? Does that make sense? So the greater the specificity, right, the higher the reaction rate. Does that make sense? Okay. All right. So the substrate specificity, and note to self and keep this in your head so that when we do the next one, this will make sense. Substrate specificity implies multiple substrates, right? That means that there's a good fit and a bad fit of substrates. And keep that in your head as we talk about the next one, which is similar but different. The next one is called enzyme substrate affinity. And before we go any further, what does the word affinity mean?
Attraction.
Yeah, attraction, how well you like something. So I have an affinity for Macintosh products, Apple products, and red shoes, right? I do. And sometimes enzymes have a particular affinity for a substrate, and sometimes they have less of an affinity for a substrate. So here's the enzyme substrate affinity. Okay, here's the deal. This is how much the enzyme fits the substrate. This is assuming one substrate, different enzymes. You and I have different enzymes, okay? Slightly different. We've got to have some of them which are almost identical because we've made it to this age, right? But there's probably some variability in our enzymes, okay? So this is an enzyme that has a very high affinity for the substrate, assuming one substrate. We're holding the substrates constant, right? And this is an enzyme with a low affinity for the substrate. Does that make sense? So this enzyme substrate affinity does not imply multiple substrates, it implies multiple enzymes, okay? Does that make sense? That's sort of the similarity and the difference between the two of them. The first one is I've got more than one substrate and the one that fits best goes faster. And now this is more likely to be true between people, not within a person, although you probably have two copies, one from each parent. That makes sense of a lot of your enzymes. This one says that if you have an enzyme that has a greater affinity for the substrate, then your chemical reaction is going to go. And that may be why that girl's way skinnier than you, just so you know, right? because her enzymes have a greater affinity for the substrate, and so it's genetics. So what can you do? Nothing. You could hate her from far. You just don't hang out with her, that's all. Don't hang out with people who are way hotter than you. Are you stupid? What? Huh?
I said, don't be the fat friend.
Unless you're Fat Amy, because I would totally hang out with her. She's funny. Okay. So we got enzyme substrate affinity, we got substrate specificity, we got, product concentration is the last one. Product concentration, I'm going to draw a better picture of that one. Okay, so this one's kind of interesting because it kind of brings the last chapter of this chapter together. Okay, so you guys know when you were looking in cells, generally it's not a single chemical reaction that gets you the product that you want. It's usually a series of chemical reactions called a biochemical pathway, right? So it's not like, oh, I do one thing and then I get the product. It's no, I do 27 things and I get the product, right? So that's called a biochemical pathway. So if we start out with like a simplified mock biochemical pathway and we say I've got A plus B which goes to C, okay? And then C which dissociates to D plus E, and then D plus E will form product F. And that's the product I ultimately want, okay? And in a cell, almost all chemical reactions are going to be catalyzed by an enzyme. So this is enzyme number 1, okay? This is enzyme number 2. This is enzyme number 3, okay? So let's draw enzyme number 1 kind of like we drew that one, enzyme number 1 over there. So enzyme number 1 fits A and B. A is that shape, B is that shape. Yes? Okay, so this is enzyme number 1, okay? Now, what happens is that there is a negative feedback inhibition mechanism that keeps you from building up too much product. And the negative feedback inhibition mechanism goes like this. When F is formed, as the concentration of F increases, it's way more likely to find these enzymes in a negative feedback mechanism. So what do you think would be logical for it to do? Turn on or turn off? Enzyme number 1. Off. And why enzyme number 1, not enzyme number 3?
Is that an F at the end?
Yeah, that's an F. Why turn off enzyme number 1 instead of enzyme number 3? Because you'd be making all of these intermediate products, which are potentially expensive to make, right? Okay, so here's how this works. Very often, these enzymes have another site, and it's called the regulatory site. It's not the active site. Okay, note to self, they do not bind at the active site. It binds loosely and reversibly at this regulatory site. And what it does is F will probably make A and B fit better or worse? Worse, right? So as that one builds up, you turn off enzyme number 1, and then the concentration of F starts to go back down again, right? And then as the concentration of F starts to go back down again, not as many of them will find enzyme number 1, and then the concentration of F starts to go back up again, right? Do you see? So it's a little negative feedback inhibition mechanism, and if you looked at it over time, what would the concentration of F do over time? What would it do? No, just over time. If you looked at it, yeah, it'd go up and down like a negative feedback, right? Does that make sense to you guys? Do you see why it's more efficient to inhibit F? All F did is, I don't know, maybe change the shape so that they look like this for a little while, right? But then when F fell off, right, then the shape fits again. And so if you look at this at an individual enzyme level, it's turning it on versus off. But if you look at it at a collective level, it's really turning it up and down, right? Because as F builds up, right, more of them find enzyme number 1 and turn it off. So that turns down the level of product concentration, right? And as you run out of F, right, then the concentration, and then the active enzyme number 1 comes back up, and so it just goes up and down. That make sense? Mm-hmm. So this is how you know, even though these chemical equations don't have brains, how do you know when you have enough of something? Negative feedback inhibition, right? Homeostasis, that's what it is. Is that good? Okay, so product concentration. So the relationship of product concentration is that often the product concentration can regulate the enzyme activity, and usually what it does is cuts off or turns off temporarily one of the enzymes. And by the way, it doesn't always go back to number 1. Sometimes it goes back to 1, 3, and 5 because it's like turning it off several different places, right? But a lot of times it'll go off. It'll go all the way back to the beginning. Okay, so that's negative feedback. Okay, now what we just saw right there is an example of what we call allosteric regulation. And it's a temporary shape change regulation that we do with enzymes because, here's the deal. We don't want all of our enzymes to be on all the time, and we don't want all of them to be off all the time. But if we just waited until we needed an enzyme to make it, can you see that that might not be a fast enough response? So enzymes have on and off switches. And if you need them most of the time but sometimes don't want them on, then their default state might be on, right? And you could flip them off. But if you almost never need them, but when you do need it, you need it quickly, then its default state would be off and you could flip it on. Does that make sense? So there's two common ways of regulating enzyme activity, and the first one we just saw an example of. It's called allosteric regulation. And what happens is you use some third kind of chemical, not something that fits at the active site. So you've got the enzyme, you've got the things that fit at the active site, the substrates, and you've got this third kind of chemical, which a lot of times they refer to as a modulator, and it fits at a site called the regulatory site, not the active site. And so watch what it does. Here's the active site right here. We'll look at the shape. When the modulator fits, it temporarily changes the shape of the active site. Does it turn it off or on? Well, it depends on what its default state was. It makes it different than it was before, right? Let's say you turned it on. I don't know. Is that good? So that's what happens with your enzymes. That's why you can have all of them readily available, but they're not all used all the time, right? So this is called allosteric regulation. Usually it's a temporary shape interaction, usually caused by like a weak ionic bond. Like it's just sort of attracted. By the way, they form and fall off and form and fall off and form and fall off all the time, right? So this falling off you sort of depend on it, don't you? You depend on it forming and falling off. So that if you have a lot of it, it's just sort of constantly finding that site. And if you have a little of it, it's not. Does that make sense? Okay. All right. So that is allosteric regulation, and this actually comes into your life in certain ways that you probably never knew. So a lot of things that we know we're supposed to eat, we know why we're supposed to eat them. We know why we need fats, we know why we need proteins, we know why we need carbohydrates. But things that you're supposed to eat and you have no idea why, like vitamins and minerals, those are not actually used. You're not actually burning them via cellular respiration or anything for energy. So what do you use them as? Interestingly, most of your vitamins and minerals, a lot of them are used as these modulators or regulators for enzymes so that your chemical reactions will not happen effectively in the absence of, for instance, magnesium or in the absence of cofactor Q10. Does that make sense? So as soon as you add those, then you increase the efficiency of your chemical reactions. And when you take them away, you decrease the efficiency of your chemical reactions, which is interesting to me because like magnesium does not end up in your freaking DNA. It doesn't end up in your proteins. Where the hell does it go? You're just like using it as a cofactor or a coenzyme, and you will not work effectively in the absence of these things. So they're often used as cofactors or coenzymes, they are, vitamins and minerals are often used like that. What's the difference between a vitamin and a mineral? Not much. They're both needed in trace amounts, like very, very small quantities. But a vitamin is organic. You have to eat another living thing to get it. Does that make sense? It has to come from another living thing. Where you could eat a carrot or you could eat a puppy, but don't eat puppies. But you could get vitamins from both. Minerals you can get from licking a rock, as long as it's the right rock. Right? They're inorganic, and you still need them, okay? And so that's a lot of what your vitamins and minerals are doing. If you are vitamin deficient and you add vitamins to your repertoire, or you start eating better, you will probably notice your metabolism going up, changing your health changing a little bit. Most of us are not vitamin deficient, but some of us are. I tend to be really anemic, and when I start like taking care of myself, my metabolism, my energy goes way up. So I get more hyper than this.
I have one question because I read like 2 books and they're like completely different. One said if you like eat a lot of vitamins, it just like go extra to your digestive system. Nothing happens. But other books said it's like damage your liver, it's damage your like. So what is the truth?
Well, you have to be very careful with lipid-soluble vitamins. Those can cause liver damage, and that's ADEK, A-D-E and K. So be careful with those. Most of the other ones, you're just going to have very expensive P unless you are deficient, okay? But also, you never want to overdo the amount of a vitamin. Some people say, oh, okay, if 1 multivitamin is good, then 4 may be awesome. Duh. You know, So yes, in large amounts, they can potentially cause damage. There's a new information that came out just recently from the American Association of Pediatrics that says our kids shouldn't probably be taking multivitamins. And I haven't read it yet. I'll have to read the research. But if you are, what they really want you to do is go get analyzed to see like what you are actually deficient in. Most of us are deficient in vitamin D because we live in Utah and there's a big cloud of goo over the top of us right now. So vitamin D, they just don't want you to supplement things that you actually don't need. Males should never supplement iron unless they are iron deficient because it can cause hemochromatosis, which causes all kinds of colon and liver damage. So there's things that you should and shouldn't do. Next time you go to the doctors, make them freaking test you. I mean, you're going to have to pay for it anyway. So I had my vitamin D tested and a bunch of my other stuff, and my iron tested. So the answer is probably individualized. However, taking one multivitamin is probably not going to hurt you. Taking 3, 5, some people, my mom made us, like, we rattled when we went to school in the morning. We took so many vitamins every day. But there are some vitamins that they have done a lot of research on, like omega-3 fatty acids. A lot of us could benefit from more of those, but it's individualized. Okay, so covalent regulation is the other way to regulate. So remember what we were doing was just temporarily trying to change the shape of the active site to turn it on or off. There's another way to do it. This one is trying to accomplish the same thing, but it's a more expensive, it's slower, but it's also sort of a more permanent or stable change in the enzyme. It's called covalent regulation. And so before we get lost in the details, I just want you to look. Here's the enzyme with the active site, right? Here's the enzyme with the changed active site. So it's trying to accomplish the same thing, only it's going to do it by using 2 friend enzymes to help. Okay? It cannot do this on its own. It takes a chemical group. Let's say that's a phosphate that's on an ATP. You guys with me? Takes that phosphate and uses its friend, this enzyme right here, to slap it onto this enzyme with a covalent bond. So this is the one we're trying to regulate, and A and B are our friends. Yes? Okay? So enzyme A phosphorylates, and that changed the shape. Let's say you turned it on. Okay? A covalent bond is a more permanent change than a weak ionic bond. So if you wanted it to stay on until I told you to go off, right? Because I said so, you can do this kind of regulation. Now, you have to turn this one off as well. So the way that you would turn it off is you have to use another enzyme usually, and this enzyme actually yanks that covalent bond apart again. Okay? So do you see that this is trying to accomplish the same thing? It's only way more likely to cost energy. It's also going to be slower, but it's going to be more stable, a more stable change in the enzyme. Does that make sense to you guys? So this is called covalent regulation. The place in which you have probably seen this is with kinases and phosphatases. Have you guys heard of kinases and phosphatases? A kinase is an enzyme that if this was a phosphate, this would be, enzyme A would be a kinase because it's phosphorylating another enzyme. Yeah? And then this guy right here would be a phosphatase, which takes the phosphate back off. So they kind of work together. Once you phosphorylate, dephosphorylate, phosphorylate, dephosphorylate. Nevertheless, what these are doing is trying to accomplish the same thing, but they're trying to do it more stably. Does that make sense? Okay? All right. So now we are going to move to metabolic pathways, which is kind of the same thing. We're going to still be talking about enzymes. Okay, so with a metabolic pathway, that is like a simple metabolic pathway, in which you have a series of enzyme-catalyzed reactions to get ultimately the product that you want. And there are a whole bunch of metabolic pathways. If you ever take biochemistry, pretty much the whole time you just learn about metabolic pathways. And they are super duper duper complicated. For instance, this is only the first step of the metabolic pathway that we're going to be talking about today. But from my perspective, I need you to be able to understand it physiologically and discuss it with me from that perspective, not biochemically. Now, if you guys take biochemistry or go into graduate school, you will have to draw every damn one of these things with all of the enzymes, but that's not today. Today is physiology. So you need to learn this in the detail that I teach it to you in class, and if you read the textbook, it'll teach you a whole lot more detail than you need to know, okay? So use your notes and the lecture as a guide for how deeply to study this. You're going to have to know it as deeply as I teach it. Okay, so we're going to start with arguably the most important metabolic pathway to you as a human, which is cellular respiration. And cellular respiration is that damn chemical equation that people have tried to teach you like 27 times, and you keep pretending that they're going to go away and stop talking about it, but they're not. And I thought they would too. I tried to get all the way through my biology degree without actually learning it, and then I realized, oh my God, they're never going to shut up, and they didn't. They never shut up. So I'm going to try to convince you before we start that cellular respiration is worth learning, because it is the whole reason that you eat and the whole reason that you breathe and the whole reason that you have a cardiovascular system is because of this damn chemical equation. And this chemical equation, for me, you do not have to balance it, okay? And you only need the summary equation, which means you need the things that go in one end and out the other end. The only thing that you need to keep track of is the numbers of ATP. You don't have to do the FAD or the NAD or any of that business for me, okay? So you are, however, going to have to know the big picture and put in some details. The big picture is pretty easy, though. So if we start with C6H12O6, because we're going to do cellular respiration of, what's this?
Sugar.
Specific? It's glucose, yeah. It's glucose. It is a sugar. Note to self, you can burn other sugars. They just don't usually make you memorize the other sugar equations. You can also burn proteins, and you can also burn fats. So you could do this all over and over again for proteins and fats, but we'll get it started with this one. So the whole reason you eat and half the reason you breathe is right here. Because if you have these two in combination, you will make ultimately you will make this. And the big deal about this is without this, you are a big pink pile of goo. Because this is what you use 60 to 70% of it just to maintain homeostasis, right? All of those reflex arcs, all of that negative feedback that we were talking about, it is all incredibly expensive. And where do you get the ATP to maintain your personhood? You get it from these chemical equations. So the whole reason you eat, the whole reason you breathe, right? And the whole reason you have a cardiovascular system is just to keep things going from one place to the other, because of course you only absorb oxygen primarily at your respiratory system, but you need it everywhere, right? Likewise, you only absorb nutrients at your digestive system, but you need it everywhere. So by the time we're finished talking about this, you should have an appreciation for why you do cellular respiration. If you start running out of things to burn, sugars and then the other ones, you'd say, oh, you will die. You're not going to die first, because just so you know, if you don't eat anything to burn, you will actually burn yourself, right? You will start eating your own fat, and that's all well and good, but then you're going to start eating your own muscle, right, to keep your brain alive. If you stop breathing, you die, but why? Why do you die when you stop breathing? Interestingly, it's actually counterintuitive. It's not even a lack of ATP, it's actually a buildup of carbon dioxide. The buildup of carbon dioxide will change your pH and your proteins will stop working and your brain will turn off, right? But you can also die because of lack of oxygen to your brain, so that can kill you first. So it depends on which one happens first. So there are whys around all of this. The water doesn't seem to make any difference at all because it just gets mixed with all the water in your body. It's just metabolic water. And then we're going to talk about quantities of ATP that you make in each individual part. So do you see, and I am embarrassed to tell you how long it took me to figure out why the hell this equation was called cellular respiration. And some of you guys didn't ever get it before either. It is oxygen in and carbon dioxide out at the cell level. Cellular respiration. And some of you guys just had an aha moment too. Like, what the hell? Why did I never get that? Okay? It is like cell breathing. It's also cell eating, right? Does that make sense? Okay, so of course you guys know there are 3 stages of cellular respiration. I'm going to approach them from a phys perspective. There is glycolysis, there is Krebs and the linking reaction, and then there is the last one, which is either electron transport or oxidative phosphorylation. They can't decide what to call it. So we're going to do them in order, and what you're going to have to do is basically sort of keep track of what goes into each one and what comes out of each one. Okay, so I call cellular respiration the most important metabolic pathway, but that's only for a human. If you were a plant, plants are cooler than you because they can make sugar and then burn sugar, right? They make it and then they burn it. So like we say we're destroying the planet and we're going to kill off everything. Just so you know, those plants are going to be fine. They can just like sit around and make themselves a cheeseburger, right? So they do photosynthesis and cellular respiration, and you, lowly animal, only do cellular respiration. So we'll start with glycolysis. Glycolysis is incredibly complicated. This is what it really looks like. There are 10 different enzymes that are involved here. There's a lot of business going on, but what I need you to do is track what comes in and what comes out. Glycolysis, the name sort of tells you what's going on with glycolysis. It's glycolysis, right? Which is a breakdown of sugar. Not surprisingly, every single sugar will go in at glycolysis, okay? If it is a complex carbohydrate, you will make it into a sugar and then you'll put it in at glycolysis. So let's look at a simplified figure of sort of what's going to go on here. Where are you? This one. Okay, so this figure is way, way simplified. So just look at what you're looking at before it gets too complicated. So notice this whole block right here represents all 10 of those chemical equations with all of the changes. And this is glycolysis. They're showing you just glucose going in, but you can burn other sugars. And then notice that you're tracking what comes out on this side, which is ATP. That good? Okay, and then after glycolysis, we will have these structures right here, or these reactions, which collectively are called the linking reaction and Krebs cycle. I just usually lump in the linking reaction with Krebs. The reason I like this figure is it keeps track of what's coming out over on this side, which makes it pretty easy. And then once you get through those two, you've got oxidative phosphorylation, but with glycolysis. Also, please notice that I told you before, conceptually, you understand that you can burn other sugars, right? Okay, but you can also burn proteins and you can burn fats, yes? So this is going to sort of show you where those things would go in. I won't make you memorize those, but conceptually, you probably need to know that you can actually make some energy out of protein as well. Okay, so let's talk about glycolysis. Glycolysis is where all carbohydrates start their breakdown. Okay, you guys probably memorized in 1610 that cellular respiration occurs in the mitochondria. That's only kind of true. The last two stages of cellular respiration occur in the mitochondria, but glycolysis occurs in the cytoplasm. And the good thing about that is that it can even happen in organisms without a mitochondria. Like if any of you guys are taking micro right now, right? Glycolysis happens in prokaryotes and things that don't have mitochondria. It's really the oldest form of ATP production that we know of. Okay, so, and you do it in the cytoplasms of your cells. In addition, it's the only part of cellular respiration that occurs in you that doesn't require oxygen. So even in the absence of oxygen, you can make a little bit of ATP via glycolysis. Does that make sense to you guys? Okay, everything that occurs in the mitochondria requires oxygen. So this one occurs in the cytoplasm and doesn't require oxygen. So it does not require oxygen, so it can occur under anaerobic conditions. And what does anaerobic mean? Without oxygen, exactly. Now, why would you want to do it? Because if oxygen is present, after you get finished with glycolysis, you can keep going, and then you're going to get boatloads of ATP. It does not produce impressive amounts of ATP, glycolysis alone, but it does get you the intermediate products so that you can keep going and eventually produce impressive amounts of ATP. So if oxygen is present, the breakdown products will proceed into Krebs and then ultimately into oxidative phosphorylation. If oxygen is not present, very little ATP is ultimately formed via glycolysis. And some cells will actually form lactic acid as a byproduct via glycolysis if they go through it multiple times. So glycolysis requires 2 ATP and produces 4, so the net that you end up getting out of it is 2, okay? So what we're going to do with this is sort of keep track of where things are used and where things are generated. So we used glucose in glycolysis, correct? Did we use any oxygen? No, we don't need oxygen. Did we generate any carbon dioxide? Nope. Did we generate any water? Nope. Did we generate any ATP? Yeah. Okay, so we're going to keep track of just the numbers. I want you to keep track of ATP, okay? So from glycolysis, we generated 2 net. Generated 4, used 2, so we're just keeping track of what's coming out. Okay, so what's the big deal about this? The big deal about this is that this pyruvate molecule that you have right there, That one is going to go through a little bit of a change, and then you can dump it into Krebs. Okay. By the way, pyruvate, glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule. So actually what you did is you kind of popped it directly in half, and you've got 2 3-carbon molecules, which you're actually going to keep track of there. Okay, so we're going to use pyruvate to dump you into Krebs and ultimately into oxphos. Krebs, this is Krebs for real, okay? And aren't you happy you're not taking biochem right now? So Krebs and the linking reaction. Just so you know, FYI, the linking reaction, biochemists would hate me because I'm actually lumping it in with Krebs. I'll show you where it is. Where are you? Okay, this little guy right here, taking pyruvate and forming it into acetyl-CoA is called the linking reaction, and it produces carbon dioxide. And then acetyl-CoA, acetyl coenzyme A, will go into Krebs. For me, I'm lumping all of these together, okay? Is that okay? And it's not actually true, but it's true enough for physiology. So as I go through all of this, which is the linking reaction in Krebs, then what am I actually going to do? Well, this is where carbohydrates would continue their breakdown if oxygen is present. Okay? If oxygen is not present, you're screwed. That's all you got. You got your 2. Maybe you can go through glycolysis again. Maybe not. This is, by the way, where fats, look, and proteins often begin their metabolism. Do you see? Proteins start right about here. Fats skip here, unless they're glycerol, which has some sugar on it, right there. Sometimes start right there or right there. So anything that's not a glyco usually skips glycolysis. That's not that hard to understand. Krebs uses pyruvate, also called pyruvic acid, which is changed to acetyl coenzyme A. Krebs occurs in the mitochondria. Everything that occurs in the mitochondria requires oxygen, so this is going to require oxygen. And you form about 2 ATP. I don't say about because I don't know. I say about because there's 2 different pathways that Krebs can go. One of them forms 0 ATPs, and the other one forms 2, so it's 0 to 2. And then carbon dioxide is, of course, formed as a byproduct from both linking reaction and Krebs. You see that? Okay, so if we're keeping track now, did we use any more sugar, or are we still dealing with the first sugar? Still dealing with the first sugar. And did we use any oxygen? Yeah. So the first time we used oxygen was in Krebs. Did we generate any carbon dioxide? Yep. How about water? Did we generate any water?
No.
No. And we generated how many ATPs?
2.
So with Krebs, we'll say 0 to 2. 0 to 2. All right. So all of this business for 4 ATP maximum, not at all impressive. Why did you go through all this trouble? Because this thing right here, coenzyme 2H, is the only thing that can go into oxphos. And oxphos will produce 34 ATPs. Okay? So this is why all the trouble. Okay? So if we go into oxidative phosphorylation, this is where carbohydrates will continue their breakdown, assuming that there's oxygen present. By the way, some fatty acids will skip everything and go right to here because they can form coenzyme 2H relatively easily. But it uses collectively and sequentially the products that came from glycolysis and then Krebs. And it occurs in the mitochondria, so it requires oxygen. And it produces water as a byproduct. Okay? And it produces 34 ATP per glucose molecule, which is pretty freaking impressive. Okay? So, and this is it right here. So we produced water as a byproduct and ATP, and we needed oxygen. Okay, so let's just go back one more time. Did we use any more glucose? How about oxygen? Yes. Carbon dioxide, we generate any more?
No.
Nope. And water?
Yes.
Yeah. And how many ATP? Okay. And so the total, 36 to 38 per molecule of ATP in the presence of oxygen. In the absence of oxygen, how many? 2. Pretty freaking unimpressive, 2. Okay, so very rarely do I have you memorize numbers, mostly because I suck at numbers. Those you have to memorize. Okay? And don't get it wrong either. Don't say 37, don't say 35, don't say anything else, because there's not that many numbers to memorize, okay? All right, so what do I want you guys to be able to do with all of this? I want you to be able to do this, okay? In addition, I want you to be able to speak about it. So I am going to, in addition to number 5, it says you should be able to write the unbalanced summary equation for aerobic cellular respiration, which is what this is, of glucose, right? And then in addition, I want you to be able to write in 3 sentences or less the significance of cellular respiration to human physiology. Okay? Significance of cellular respiration to human physiology. Remember that you are writing for science, so no adjectives or adverbs. I don't want to hear about cool. I don't want to hear about something's pretty or awesome. Do not write that when you're writing about science. Also, do you think I'm going to give you credit and say if you say you would die without it? You knew that before you got here, okay? So you need to give me some details, preferably something about homeostasis, preferably something about reflex arcs, preferably something that gives me some detail as to why this is happening, okay? So ideally, you would be able to, in 3 sentences or less, explain it to a very bright 12-year-old, right? Why the hell does this happen? Okay? And that's what I want you to be able to do. Just work on something that's actually true and has some content in it, okay? You know that 60 to 70% of the ATP that you generate, you actually use just for homeostasis. Okay, so we will stop there for today, and next time we're going to talk about burning other carbohydrates. No more chemical equations, but burning other carbohydrates, burning proteins and fats. No quiz until Friday at the earliest, and perhaps Monday. I haven't decided yet. Hi.
Hi, our class starts at 1:30 today.
1.
1?
You need some sleep. Wake up.
I know.
You need some caffeine, my friend.
Yep.
So Tuesday in the
South City campus.
and it's like office.
It's hard to find. Yeah, SEC 105. It's in the new building, the little glass office.
So in this case, it's better for me to find it here, right?
Yeah, because I'm right after this class on Wednesday.
Wednesday after this class.
Right after this class, so you can just walk over with me if you want to. Yeah,
And it's like okay.
Yeah, I'm just right over in the...
And okay, I will very quick because today we will like fight with my husband, and he said like, hey, go to your physiology teacher and ask. And I'm like, okay. So the problem is my youngest son, he got sick, and he has like run nose. I asked him to go in the store and buy the medicine for him. So when I wake up in the morning, because I asked him 3 a.m. to go and buy because he was not sleeping well, he bought the allergy. So he